I want to get a part of that, then how that will secrete H plus and OH minus. I mean H plus and Cl minus, and I'll put a separate page for that. Okay, this is a stomach cell where I mentioned before H2O, that's water plus carbon dioxide. Okay, this is the stomach cell, stomach and parietal cell. Parietal cell, okay, where this carbon, they are secreting a hydrochloric acid. Okay, and here it will form H2CO3, H2CO3 carbonic acid. It is called carbonic acid. Carbonic acid. H2CO3. This is very unstable, this one. Can either go back directly back here, H2CO2, or it can disintegrate to HCO3 minus, that is a bicarbonate ion, and H plus, that's another proton. Okay. The enzyme which will facilitate here is carbonic anhydrase. Carbonic anhydrase. This is the enzyme which facilitates the water and carbon dioxide together to carbonic acid. What you drink soda, what is that? Carbon dioxide and water. Whatever you are drinking in the soda is nothing but carbonic acid. So what you do whenever you just open your soda and keep it open, after a while you cannot drink it. Why? Because the carbon dioxide is gone, and then because it's a reversible reaction. When the oxygen present in the air, that will dry, and the carbon dioxide, when compared to water and carbon dioxide, that's very unstable. There's no enzyme in that one, carbonic anhydrase. But if you open it, the gas will go up, and then you won't get anything on pot. Okay, so you have a high concentrate content of carbon dioxide to keep it in a carbonic acid in an environment. Okay, and if it is warm, it facilitates the elimination of carbon dioxide too. Okay, that's why you have to keep it in the fridge a little bit. Okay, but when compared to outside, it is not. Okay, so that's a very simple thing, you know. But the same carbonic acid is secreted, it's present in our individual cell, that is on parietal cell, where H plus and HCO3 minus bicarbonate disintegration is taking place there. This bicarbonate now will go out, okay, in the transporter, an antiport mechanism, where a chloride minus ion, this is a bicarbonate minus ion will go, and then a chloride minus ion can get import into the cell. So once it comes into the cell, okay, and it will go out to the cell. This is the lumen where your food particles are coming here. So this chloride now passes through here, Cl minus. Okay, this is a chloride channel. We call it as a channel mechanism. Chloride channel. Okay, this H plus ion, this will move outside too, and there is a transport mechanism where H plus K, that's on a potassium, which is coming in, and the one hydrogen is going out. So now you get in the lumen H plus and Cl minus, and to form hydrochloric acid. This one and this one to form hydrochloric acid. This stomach. This is a very simple phenomena, or the principle which involved in hydrochloric acid secretion in stomach. Do you follow that? So in simple terms, here from this one you can call it as a serosal side, serosa, and this is the mucosal side where the stomach mucosa. So here you need, but what happened? The chloride, which is not present inside normally, but at the expense of the bicarbonate pushing out and more chloride in. Okay. So now I want to give you a small scenario. If you drink soda, what will happen? What will happen? Which will facilitate? Will it facilitate more of what?
Carbon.
Just a second. If you take carbon dioxide, right, and water, right, you are taking this carbon dioxide and water, right? That's not soda, right? That's what you are drinking, right? So when you drink this, you are activating the carbonic anhydrase reaction, because more of that. And if it's more, it will drive the forward reaction. Forward reaction means carbonic acid will be synthesized inside your parietal cell. It is absorbed, it's going in more of carbonic acid in your stomach. Fine. And then that will disintegrate in bicarbonate ion and H plus ion. And the H plus will come out as a H plus, as a potassium sandroot. What will happen when you take excess soda? The lumen, whatever the lumen potassium is depleting it out. So potassium is going in. So you get at the expense of hydrochloric acid. And then here, when the bicarbonate is there, it will push there, and then from the serosa side or from the plasma, the blood, you get more chloride is going to be there. But your blood is going to be more of bicarbonate minus ion, more of HCO3 minus is going to be there in your blood. That's okay. H plus, sorry, Cl minus will come out at the expense of this channel. Chloride channel is deactivated, and now you get more of hydrochloric acid. So when you drink soda, that translate mean you are secreting more of hydrochloric acid in the stomach. Am I making sense? If you drink more soda, hydrochloric. So if you drink soda on an empty stomach, already your stomach is empty and it needs some food. Okay. At that time, if the empty stomach, if more acid secretion, what will happen? That induces a peptic ulcer, right? Empty stomach, if you drink more soda, what will happen? You are adding more fuel, more chloric acid, hydrochloric acid in the stomach. And also you are increasing the bicarbonate ion into the serosal side. Okay. So that is a bit dangerous. You cannot keep it. Okay. The same thing when you drink soda with aspirin. Have you ever tried that? That is also a bit dangerous. Not just like an alcoholic effect. Don't do it. Don't try. Okay. But first you drink and then add aspirin and then drink it. What will happen is the acetyl salicylic acid, and then this carbonic acid, this one will increase, and more of bicarbonate will go into the bloodstream. That gives into your brain receptors and then make as if you have drinking alcohol. See, in those days they banned alcohol, not to drink. The kids and college students, and they used to have a fizzy drink and with aspirin and then make a same effect on that part. But that dangerous. Sometimes it affects the heartbeat and everything. So it is not advisable. But that's one other thing. Okay, you should remember. So if you drink empty stomach in a soda, this is another effect which is going to happen. So in coming to our digestion part, what this hydrochloric acid will do? It will increase the H plus or the proton concentration in the lumen, where you have more of proteins need to be digested by what? I'm just drawing another one. In the stomach, the protein digestion will start, right? Protein, and that is by chymotrypsin and trypsin. Okay. And then it is going to be a polypeptide. So this pepsin and trypsin, they have a proenzyme called proenzymes.
Sir?
Yes?
We really can't see the images, so it's kind of blurry.
Okay, I'll
is there
change
anything
them.
you can do with the lighting?
Wait a minute, let me see. Again, I put the lighting on.
Maybe like a thicker Sharpie. There you go. Well,
Could you see now?
um,
No? Could you see? Proenzymes, trypsinogen. Okay, this is a proenzyme. Could you see now from Syncorange?
what's the word at the top?
Chymotrypsin. Chymo, CHYMOTRIYPSIN. Chymotrypsin, this one. And this is a trypsin. Okay. So these are enzyme, proteolytic enzyme. Okay. And this is a trypsinogen, and the another one is the chymotrypsinogen. Chymotrypsinogen. Okay, these are the proenzyme. These are the proenzyme. This is the proenzyme. So for this proenzyme, we need hydrochloric acid here to here. HCl. So this acid will cleave the chymotrypsinogen proenzyme to the enzyme, active enzyme. This is very active. And then hydrochloric acid will convert the trypsinogen proenzyme to active enzyme. This proenzyme is inactive form. Could you clearly see that, or still is a problem? Sync orange?
We're just trying to figure out what's in the up left-hand corner.
Chymotrypsinogen.
Thank you.
Okay. So the inactive form of the enzyme to active form. When you do this, you need hydrochloric acid. So that's the part. Number one. Number two, the hydrochloric acid at the low pH, the pH is going to be around 2, pH equal 2, and that is precipitate protein. So, hydrochloric acid, precipitate protein. Okay. Oh, okay, fair. There you go. Precipitate proteins, and the precipitated protein will be acted on by these enzyme, active enzyme, trypsin and chymotrypsin, and thereby it is going to be converted into polypeptides. So the role of hydrochloric acid, there are 3 functions of hydrochloric acid. Okay, I'll write it in separately. Hydrochloric acid. Okay. Function. What are the functions of hydrochloric acid? That's what we are going to see. Number 1, precipitate proteins. Whatever the protein you are eating, you are going to precipitate it out. Okay. Number 2, and it gives proenzymes to active enzyme. This inactive form, active enzymes. That's a chymotrypsinogen to chymotrypsin, trypsinogen to trypsin. So these are proenzyme, which is the inactive form. Inactive. Number 3, since it's the low pH, okay, it acts on innate immunity. It helps innate immunity. Meaning, suppose if you drink some water or any drink or food containing bacteria or any little fungal spores and everything, that is being killed by hydrochloric acid. Follow? So these are the functions of hydrochloric acid. Now, what we have studied so far. Stomach, we have a parietal cell, which is responsible for secretion of hydrochloric acid. That's fine. H plus ion hydrochloric acid. And chloride, yes, everything is fine. Now we are studying the functions of the hydrochloric acid in terms of digestion. Number 1, we did with the role with the hydrochloric acid will precipitate protein. Whatever the protein which you are eating it. We're eating meat or vegetables or anything, whatever the protein. So the protein is getting precipitated by this. In a lab, have you done that one? Any cell homogenate, and then you are adding some, not hydrochloric acid, sometimes you add glacial acetic acid or phosphosalicylic acid or TCA, trichloroacetic acid. You have done that before. So that you are increasing the hydrogen ion concentration, and thereby the proteins, which is amino acids and what is called? There's a polypeptide chain, right? The peptide bond that getting aggregated together to form an insoluble form. That from soluble protein to insoluble because of hydrochloric acid secretion. That is the mechanism which is present for the precipitated protein. Why we need to precipitate the protein? Then only the enzyme will, this active enzyme will act on to the protein. If it is there just like in a soluble form, it is not quite possible because the active site should be fit into that so that the protein can come and sit so that it can digest the part. So active enzyme. And then this is nothing to do with the digestion, but it is an immunology part. That's an innate immunity where it prevents any bacterial spores or bacteria to survive in the stomach. So and thereby it helps. Do you follow the hydrochloric acid function? Yeah, you will get an exam question on that part. Okay. Now, the next one, after the digestion of the protein, then food will enter into the next part is intestine, small intestine, okay? Small intestine. The small intestine, it is going to be divided into three parts of the small intestine. Duodenum, okay? And the next part is jejunum. And then third part is ileum. Okay. I love the small intestine because I did extensive research on the small intestine, is the animal model as well as in the human subjects, and then we have done some more work. So it's a quite possible then we can study the transport mechanism, everything non-invasively in human itself. Okay. We'll study about the duodenum. What is the function of the duodenum? Duodenum is a secretory function mainly. Secretory function. Secretion of what? Enzymes. So duodenum is a secretory function of the enzymes. Okay, and it also hosts some other thing, enzymes from pancreas and liver. So pancreas and liver organs, and they also put forth their enzyme to the duodenum as a secretory function. But the pancreas will produce lipases, and liver will produce from the gallbladder, okay, and gives into bile, And these enzymes will help again further digestion and absorption. Okay. And the jejunum and ileum, this part of small intestine, mainly their function is absorption. Okay. So the secretion of these enzymes, as well as the secretion of hydrochloric acid, they are regulated by hormones. So that part we will see on our next lecture, but I want to finish this absorption small intestine function now. Do you follow now? What we are going to split into all these parts again. Okay. Again, we will go on to the next part, is in the what happens in duodenum. What is the function of the duodenum? Secretion. Secretion is the function. Okay. The epithelial tissue, epithelium in this duodenum epithelium, they have got a lot of enzymes. They are host of enzymes secretion. Host of enzymes. Okay. And they are coming from, these enzymes is coming from liver, that's I mentioned before, bile, that's coming from gallbladder, and this by contraction of the gallbladder, what you get the bile is pushed into small intestine, coming in the intestine. So the purpose of this bile salts, and also the function is alkaline, A-L-K-L-A-I-N, alkaline. This will neutralize the pH. Remember, when the stomach, the digestion occur at the time of the stomach, pH is 2, right? So this is the acid range of the pH because of hydrochloric acid. But as soon as it leaves the stomach and enter into the duodenum, you cannot keep as an acid pH because the enzymes we are going to discuss about the lipases, they need neutral pH. They cannot work in the low pH. How many of you done biochemistry? Have you done the enzyme kinetics? Depending upon the pH, you need an optimum pH for an enzyme reaction. The enzyme cannot act all the pH, meaning it cannot act 1 pH, or it cannot act 2 pH, it cannot act even 10 pH, a highly alkaline pH. So each enzyme will have in a specific pH, specific environment to activate. So the stomach is an acid environment, more of hydrochloric acid. You get that enzyme, the trypsinogen, and then trypsin as a chymotrypsin and chymotrypsinogen. Those can act in low pH, but not the lipases. Mind it, lipases and carbohydrates, which is present in the small intestine, they need a neutral pH. For the neutral pH, what the food, digested food enter, and that also will get some neutralization by the bile. So the bile will secrete more of like a buffering capacity. It will provide more of hydroxyl ions. So that ions will help them to neutralize the acid pH to increasing the pH. Neutralizing means we to a pH 2 to probably 4, 5, or 6, around that range, so that the enzyme can activate. So you need to know whenever the food, after digestion food enter in the duodenum, it is neutralized with the bile. Okay. Then alkaline and neutralized pH, and it also produce the bile salts. What is the bile salts will do? It will help on emulsify fat. Emulsify fat, that is on the lipids. Fat and lipids. So whatever the oil, the fatty substance, highly saturated or unsaturated, doesn't matter. Any fat which you are taking it, on. So in your stomach, it won't touch anything, the acid, the acid environment. But as it is mixed with the oil, okay, and then it enters into the duodenum where it activates the secretion of the bile salts as well as the pancreatic lipases. And the lipases will act on these lipids, but it cannot act just like that. Why? Because do you know about any oil and water? Can it mix together? What normally you do, you in your shirt or your fabric container, you have some oil spill or anything, so you apply. If you dip it in water, will it go? So what you need to do? Greasy stuffs. Yes, come on. Detergent, correct? Soap, detergent you do, right? In your detergent at home as well as there. So in our body is also having this detergent, and that is the bile salt. So bile salt, imagine bile salt and bile, it is a soap, a detergent in our system. So that will help the oil and water combination. So when you do that, the bile salt will saponify, emulsify, making a soap, making a froth, and you have a foam, right? So at that time, the lipases can act on that. So this is the oil or the fat and the water in the presence. So you need here bile salts, the cholic acid, glycocholic acid. There are different type of bile salts out there. So the bile salts will go and bind and saponify and emulsify, and then after this, then you get the enzyme will react on that one. Enzyme lipases, pancreatic lipases. The lipases now can act on this oil, and then the long chain fatty acid. I'll draw the other one. Suppose this is a triglyceride molecule or the long chain fatty acid. Okay, this is a fatty acid ester. So what happened here? You get more of the bile. I'll put a drawing in a separate color, which will get. So it will bind here. So this blue color, which denotes the bile salt, and thereby it will mix the oil and water interface. So you get more of the saponification or emulsification. Now the enzyme can come and cleave this wherever it binds, and then you can cleave that one. So you will get a free fatty acid, and then you get as a glycerol whatever you are eating of the fat. So the fat is being digested now in the duodenum. You follow? But still, what you get is the product of this. is the free fatty acid, glycerol, and plus the bile salts. So free fatty acid will absorb and glycerol is absorbed. Bile salts is not absorbed in the jejunum but in the ileum. I'll come to later bit. Okay. If you follow any question, Sugarland, Cinco Ranch. Okay. Yes, go. No? No questions? Okay. Okay. So what happened here in the duodenum, just to finish the duodenum, you got the excretion properties. Okay, excretion of the pigments, the bilirubin, pigments, and from this pigments is coming from hemoglobin and also from the cholesterol. So yeah, am I getting it now here? Okay, cholesterol. And also the toxins. So duodenum is the place where the digested part of these pigments and toxins from the human, they are coming from liver, okay, detoxify and toxify. They are also excreted into the duodenum. Also pancreas, or the pancreas will do a host of enzymes, and then here it will also put, as I mentioned before, is the enormous amount of carbonate ion, and this will help neutralize the pH. Imagine, just I want to recollect my first part of the lecture, which I mentioned before. Could you recollect this one, this drawing, where the parietal cell will give more of the H plus ion, and then bicarbonate is pushing towards the serocell side. Okay, that increasing the concentration. Now that will help in neutralizing the fat. So the one which I just mentioned before, here it is, neutralizing the bicarbonate ion. So in turn, the soda whenever you drink, and that will also help to digest your protein as well as the fat. In the sense, it can direct the more of bicarbonate ion which is getting into the serocell side or into the blood plasma. Again, that will be used in the next part, in the duodenum or into the pancreas, and to secrete it. Number one. Okay. If you, yes.
I'm not clear on what the duodenum does to pigments and toxins. You said these come from the
It's coming from liver. So the duodenum is a place where it receives the pigments and receives the toxin from the liver. So I'm talking about the digestion part, but this one is also, I'm talking about the duodenum, general function of the duodenum to receive this material from the liver. You follow?
So it goes from here, it goes to the large intestine and gets...
That is correct. That is right. It goes along with that one, along the pathway. Correct. Yes. I'll put it that way. If you have a block in the gallbladder, in the liver, okay, what will happen? Instead of secreting the bile, okay, so the bile salt and everything will form a beautiful crystal inside, and gallstone will be formed, and that stone, just like any stone, when you can see that one in outside in a small pebble like thing, so you'll get a different color because of the bilirubin and other things, the green color and then red color, yellow color, orange color, like a crystals will be formed in the gallbladder, and the the bile duct will be blocked by this one, and that is the reason for the gallstone formation. When the gallstone, then it's a terrible pain onto the liver, onto the stomach, into the abdominal cavity, so they have to remove it immediately. It's an emergency surgery they normally do if it's a bile salt accumulation and stone. So, and if it leads to obstructive jaundice because of the pigments which is not secreting into the duodenum. These pigments will enter into the bloodstream. That gives a color, yellow pigment because of the bilirubin pigment onto the blood. And then you will see on your eyes and your hand and touch and sweat, everything is going to be in a yellow color. That is jaundice. Okay, this is the obstructive jaundice, they call it. The obstruction due to the bile salt accumulation in the gallbladder. Okay? So the duodenum is the place where the secretion which is taking place. Will is it okay? Am I explaining correctly? Okay. The pancreas is the one which will neutralize the acid which is previous one. That is in the stomach. I told you stomach is the low pH, and that's coming down onto the bicarbonate ion to neutralize in the duodenum by the pancreas, which will produce more of bicarbonate secretion. That is also coming from duodenum. So now the duodenum that receives the pH is neutralized pH. Do you understand that part? This is one. Now let's go on to the absorption level. Okay. What happened in the absorption? Last a couple of class ago, we discussed mostly on the different type of transport mechanism. So here we will see how just mention anatomy of the small intestine. Do you know about? We discussed in earlier also. Our intestine is surface area is something like what is called a tennis court. Have you seen a lawn tennis court? Yes, that is a surface area. Each and every one of us will having this tennis court in our small intestine. That is the surface area where the absorption is taking place. Imagine, not like a ping pong, table tennis. No, it is a field. What is called it? Lawn tennis. Yes, the lawn tennis you call it. It's a tennis. Okay, good. So the intestine, which is going on to like a folding like villi, right, and then each one will fold again to the microvilli. These will go, and then from this end also will folding like this. So it has mostly on villi, and then villi is divided into microvilli into brush border membrane. Something like the folding of this membrane, something like if you just think like this, okay. That's coming out here, and then going over here. So these are the cells. So each cells here, if I magnify it here, so you have got this microvilli and brush border membrane here, brush border membrane. So this is the place, one single cell. So you have a villi and then folding and folding and folding, so that we spread throughout, you will get a tennis court level. Okay, what happens here into the folds, and the one part is the villus. Okay, so I'm just drawing the one cell onto the brush border membrane onto that. So what you have here, you have a cell which is okay, the individual cells. Okay, so you have a nucleus. So in between you get you have a blood vessel which is going on. Okay, and then you also have neurons, the lacteals, tubules, everything. So this epithelium here, this part of this epithelium, you get amino acid, this epithelium, amino acid and sugars, they are transporting it. Okay, they are, because there's a specialized transporters are there, and that will transport into it. And here you get the blood vessel. So it will transport into the blood vessels. Okay. And from this blood, where this blood will go? It will go through the body, the capillaries, to liver, and then liver to body. So that is the pathway which will go in. The another one, which is a dark one, which is, as I mentioned before, is the lacteal. And this lacteal, which will act as a lymph system. Okay. And then it will go on to the blood, and then go enter into the liver, and then to the whole body. So this, the fat, will go directly at the digestion of free fatty acid. That will enter into the lacteals. This one, the fat will go enter through this the diffusion process onto the lacteal, and then the blue one, which I mentioned, the blood vessel, that is on the amino acids and sugars. So there are two pathways you should remember here. One is the sugars, digested sugars and the amino acids enter through blood vessel or the capillaries. Number two, free fatty acids, FF, free fatty acids, that will enter into as a lacteals. That's again, it will enter, yeah, it's also a vessel, but it's not a blood vessel. So it will go on the lymphatic vessel. I put a lymphatic vessels on lymph, and then it will enter into lymphatic system to the blood, and then it's entering the blood and liver. The blood vessel directly enter into the liver. So that's the difference. So fatty acids absorption, a little bit of a different route when compared to the sugars and amino acids. That you should understand. You follow that? So now let us see how the individual sugars will be transported across. Okay? I'm drawing a microvilli where these are, you have a brush border membrane like this. Okay, here. So here you get a capillary endothelial cells. These are the endothelial cells. Okay, these are capillaries. So microvilli, these are the microvilli. And what happens here, this is one cell, okay, this is the another cell. In between that you have an intracellular space, this one, call it as intracellular space. So the amino acids, and here you know there's a junction in between the tight junctions here, in between two cells you get the junction. And you have an amino acid as well as a sugar or digested monosaccharides. They can enter directly through this. There's a transport mechanism, active transport mechanism. Active transport. Some of them are the passive diffusion as well, transport or facilitated diffusion, some of them. So it will go enter through this transport. And then the fat or the fatty acids, that will go through this but enter into this, okay, where the intracellular, where you enter into the lacteals. And the sugar which is going in, and that will go directly into the capillaries. Yeah. Go through the intracellular space and then capillaries. And then it will enter into the lacteals. Lacteals transport is different, lacteals. So you should remember, am I making sense? Okay, good. So you get the fatty acids transport is one side, and amino acids is also an active transport, and this fatty acid is going through the lacteals by diffusion. Diffusion is the process where it will go. Okay? You follow? No. If you see the cross section of the small intestine and the cross section of the villus, I'll show you, or in your textbook also you will find some of the pictures. But just I want to explain only the mechanism here. Like the amino acid, I'm just drawing a larger picture of a single cell, and then in between the cell, okay, what happens? And then here, this is the tight junction, and there is one cell, this is the another cell. Okay. There are specific co-transporters are present. Again, amino acids and sugar and nucleotides. Nucleotide. Okay, nucleotides. Okay. They are the specific transport mechanism. You will get the sodium ion and here the amino acids which is coming up. So these amino acids and the sodium, they call it as a co-transporter, co-transport mechanism, which can go in. And then at the brush, this is the apical membrane and this is the basal membrane. Another transport mechanism where the potassium, which is going in, and the sodium is coming out. The potassium in and sodium is out. Okay. And another one is the facilitated diffusion. This is called the from inside to outside where the amino acids which is coming out. Okay. When you do the sodium and potassium on this exchange, you get the sugar will be transported into the space. So if you remember that how this amino acids is being co-transported from apical brush border side into the cell, and this is inside the cell, and then it exceeds the cell through the transporter mechanism, amino acid, the facilitated diffusion, because this is the protein, the round which I mentioned here is a protein. Protein, we call it as facilitated diffusion. So these transporters, we got the sodium amino acid transporters and different amino acids. You have leucine transporter, valine transporter, different amino acid. They have different type of transporters are there. Same thing with the glucose transporters, fructose and galactose. So individual sugars, they have individual transport mechanisms are there. Okay. If you have disaccharides, like a sucrose, if it is not digested, and that will hold more of water there, and then that will push round into the colon. If it is undigested sugars, that is a bit dangerous. Okay, that can be transported into the blood and blood to the kidney and kidney to the urine. So if the people who cannot digest the sugar, then the urine will have a sugar. Will have sugar. For example, in Eskimos, Eskimos who's living in Alaska and other places, Greenland, Iceland. Those people, they don't have a sucrase enzyme. The enzyme they cannot digest any sucrose, whatever the cane sugar, whatever you are eating it. If they eat a cake or sugar and their urine is full of sugar, they are not metabolized in their system, and it is excreted into the blood and blood into the kidney and kidney to the urine. It's very simple. So we use that technology. If anything happens in the stomach or the intestine, there is an inflammation where the sucrase enzyme is deficiency, the patients also will secrete more of sugar. Also, some kids, they used to secrete sugar, essential fructoseuria. When you see that urine will have full of ants will come and eat and all the things. It's not a diabetes condition, but it is an entirely different one as a sucrose urea. Sucrose in the urine, you call sucrose urea. Okay. Anything else? So that's one part which I want to explain to you about the sugar and nucleotide. Also, you have a DNA, nucleic acid, or RNA, whatever that contains the nucleotides that also transport the same mechanism with this one, nucleotide transporter, an active transport mechanism. Okay. I have done some work on the absorption and the different pathology, so you can go through my electronic reserve and find the papers, and you can study on interesting papers in 82, 83, and then recent one is around 1996 and 97 also, some other papers. You can study the human as well as the animal model. Okay, so we will get to later part of the class. Okay. The next one is the fat digestion. So what I'll do, I'll give a break. Come back another 5 to 10 minutes, and then we will go through that. Okay. What happened in Cinco Ranch? Are these people are there or they're gone? No exam, they're gone, is it? Are they there?
We're all here, sir.
They are okay, all there. I'm sorry, I couldn't see you. So come back. Sorry?
I think one girl went to the restroom.
Oh, okay, okay, good. Okay, now you got fat. The fat droplets, as I mentioned before, the fatty acids or any of greasy stuff, what we eat, saturated, unsaturated fat is being digested using the pancreatic lipases, and that forms like a fat droplets. Because when you do mix with oil and fat, oil and water, what will happen? You have droplets, like the same thing as the fat droplets, and that is being acted with bile salts. Okay, and they form like a micelles, M-I-C-E-L-L-E-S, micelles. Okay, so when I draw here again the another cell, a part of the cell, and this is another cell, neighboring cell, I put it that way, and this is being by tight junction. They are tightly closed by proteins. Okay. So the micelles will, micelles or micelles, will diffuse in diffusion, okay, into membrane, diffuse into membrane, and they form, I put it like in a endoplasmic reticulum, I put it like a ER, like an endoplasmic reticulum. As soon as it diffuse, the membrane will have a folded endoplasmic reticulum like that. It will go inside the cell, right? So it will go inside endoplasmic reticulum as the micelles. I put M and then M. It will go like this. So it goes, it travel through the endoplasmic reticulum, and then finally it forms a Golgi body here at the end, a Golgi vesicles. And then what happened, this Golgi, which is by exocytosis, you know about the exocytosis mechanism, which we talked about it. Exocytosis. In the basal membrane, this is the apical membrane, and this is the exocytosis. This is the M, the micelle, whatever the things which is coming out, the fat droplets of the 5 bile salts, and everything, and the digested free fatty acids, everything which is coming here, and then it forms like a droplets, and this is going to be in the endoplasmic reticulum. What will happen in endoplasmic reticulum? You have add proteins. Okay, and especially the lipoproteins, add proteins, and it forms with the micelle and free fatty acids and everything put together, and then it forms like a chylomicrons, and this one, which is exocytosis from the intestinal cells directly into the lacteal, and this is present in the lymph system. Okay, and the lymph system to blood, and then blood to liver, and then liver to whole body. Okay, let's make a little bit up. This process where you get the bile salts, which will aid in to form the micelles. Once the micelles is formed, the bile acid is not entering into the intestine directly, but the free bile acids is going to be floating around, okay, and that is going to be absorbed in the part of the ileum. So we are still in the jejunum, where the fatty droplets is being absorbed in the cells, and as soon as it enters the cell, it enters into the endoplasmic reticulum, where the micelles, you get an additional protein, that's on lipoproteins, which you can add to it when it passes through this, and then entering the terminal part of endoplasmic reticulum, you get a Golgi apparatus, or Golgi vesicles will be formed, and these vesicles, which is being pushed out of the cell into lacteal system, or the intracellular space, and then enter into the lacteal system by the process called exocytosis. So we already studied about exocytosis in our previous class, about the production, the pushing of the vesicle outside the cell, or the exocytosis process. So now these vesicles are these micelles with chylomicron. They are called it as a chylomicrons. Chylomicrons. And that is going to be transported to the blood through the lacteal and lymphatic system, and then it is going to be on the liver, and the liver to the entire body. Do you follow the how the fat is absorbed? This is the fat absorption. What will happen if you have excess, there is no bile salts? What will happen? No bile salts. No bile salts? As I mentioned before, is if the obstructive jaundice or the gallbladder obstruction of the gallstone, no bile acids is going to be there, and that bile acid is not available to make a saponification, to make a detergent reaction. So the fat is not going to be digested, and it is going to push to the colon, and then it's going to be excreted out. The same phenomena, what they are thinking, that if you take instead of bile salt, some of the fat is not a burner, but they used to add some of the, to reducing the body weight, or they used to add some of the dietary supplements. Have you ever heard of that one? There are several things which is not being absorbed, the fat absorbent like thing, whatever they eat, fat, it will come out. And so there are a lot of things is going on into the market, but you should remember that the bile salts, which is added here, and excess, if you eat excess of fatty food, you are likely to secrete, you are giving more task of bile salts and biles, that secretion. And this excess of bile salts now is going to enter into the next system, which I am going to draw here on the ileum, okay? And this is a one cell, this is a neighboring cell, okay? The nucleus, and you have a blood vessels which is going on, okay? So there is a transporting process, we call it as a bile acid transporting protein, otherwise bile acid transporter, BAT. So if it is present in the ileum, I put it ileal bile acid transporter, IBAT, I put it IBAT. Okay. So the IBAT, what this will do, it will capture all the bile salts which is floating around after the absorption of the fat. See, before. So this process, it will helps the micelle formation. Once the digestion which is occurring in the intestine, then it is being transported, and this leftover bile salts, which is being absorbed by an active transport mechanism here into, which is present in especially only in the ileum. It is not present in the jejunum or duodenum. Do you follow that? So because when you see the scenario of the small intestine, I'm just drawing here, right? Just a stomach, and then the duodenum, and then you get small intestine folding, and then ileum part, and then you get the started colon. So the terminal part of the small intestine is the ileum. Terminal part of the small intestine is the ileum. So after the absorption of the fatty substance, after digested fatty acids, these bile salts which is going to be pushed to the ileum, and then ileum is having a specialized transporting protein which is called the IBAT membrane protein that's transporting the bile into the cells and then into the blood, and then from the blood it will go into the liver, and then liver it will go directly to cholesterol synthesis, because the bile salts is being produced from cholesterol. So this will come as a entero, and then it's a liver, so it's a hepatic. The circulation of the bile salt, we call it as enterohepatic circulation. I put it in a different way like this. So liver, that will produce what? Bile salts from where? This liver will produce the bile salts from cholesterol. CHL, I just condensed the abbreviation cholesterol. So this cholesterol, this bile salts will be useful, secreted in duodenum, and then with the pancreatic lipase, it helps to digest fatty acids or the fat, and then the leftover bile salts, okay, they absorbed or I put absorbed in ileum, and then it is being shuttled to liver again. So here in the intestine we call it as a entero, and then liver we call it as a hepatic. So the shuttling of bile salts from liver to the intestine, and then intestine to back to the liver, and this process is going on a circle. It's a circle. Do you follow that? And this process we call it as enterohepatic circulation. So you should remember clearly, because whenever the excess glucose and amino acid, whatever we discussed earlier, so excess glucose will go into the liver to the glycogen storage, and muscles the glycogen storage, and for the other cells for the energy. Amino acid will go into the liver and also the other part of the body to produce another polypeptide, and for the building blocks of our own proteins and different hormones and everything, that protein will be useful. Now the bile salts, which is also absorbed at the terminal ileum, and that goes into after absorption, bile acids, where it will go? It will go into bile salts too. After absorption, go into the bloodstream. Did I mention before? Yes. Here, see, bile salts after transporting will go into the blood. Then after the blood, it will go into the liver. Okay, and the liver again, it will be converted into cholesterol. The liver, it will produce into the cholesterol here, and then the cholesterol that will produce the bile salts. So to control the cholesterol level, what you can do, what you can suggest now on this part. Suppose I want to reduce the blood cholesterol level going down because of the cardio, just with a problem, so I want to reduce the cholesterol level low. HDL cholesterol is good because that's a high density lipoprotein. But the LDL, where the more cholesterol is going to be there, high level of low, is that right? Low density lipoprotein, LDL. Okay, so this cholesterol should be reduced. If I want to reduce this cholesterol, what I have to do, I have to stop this enterohepatic circulation of bile salt. Otherwise, I have to take some cholestyramine or something like, where that will remove the intestinal, this part. It will block the bile acid transporter, and thereby to remove all the bile salts away from the, it prevents the reabsorption. In other words, the enterohepatic circulation is going to be blocked by different drugs and other mean, and thereby you are reducing the load of the bile salt into the liver. For suppose you said oats, if you take oats and you can reduce the cholesterol, what the oats will do? The oats, the contents in the oats, that will remove these free bile acids, and that will remove completely and then pushing to the colon, and it is not available. These liver, they won't get any of extra or enterohepatic circulation of extra bile salts, and thereby the cholesterol level will go down. So cholesterol, so whatever the cholesterol already there, it will be resynthesized into bile salt. There is no more input, no more feedback, no more supply of the bile salt to the liver through the enterohepatic circulation, because the bile salts which is going from here in the ileum, that is being removed by the oats or any other means. So that's why they said to reduce the cholesterol you can take more of oats. That's one of the mechanisms which they are talking about. Okay. So now, do you follow now? Yes? Get it? So when I did my work, one of my paper is there on the bile acid transporter, because we cloned the gene which is responsible for the protein, and we have studied on to that ileum part in the hamster as well as in the human. And this mechanism we did with a protein, with the immunologically identify the protein into the rat as well as into the human as well as in the hamster. So that's what part of my work in Seattle, and the bile acid transporter excessively on that. So we have some publication, you can check that too. Okay, next part is in the colon. Now we are talking about the colon. Okay, what the colon will do? The colon is something like a recovery station, recovery of water, because what the secretion, intestinal juices and gastric juices, all juices will go into the colon, and after the digestion, the excess water should be reabsorbed, recovery, and also the products of secretion. Water from the product of secretion, and especially in terrestrial animal. Terrestrial animal. And it forms as a fecal formation. So that's in the part of this in the colon. Okay. How it will happen here? In the water transporting mechanism, there is a aquaporin. Aquaporin is a protein that present in the colon that helps and reabsorb water from the colon into the system. Okay. So in the total of the digestion part, the bile acids, if the bile acids it is not reabsorbed, okay, if it's the excess of bile salt, or if you take excess fat food, and you are likely to get more of a chance of getting colon cancer. The reason, it is not due to the fatty acid as such, but the bile salts which is overloaded, and that bile salts which is not being here, because the capacity of bile transporting protein is minimum. So when it is reduced, it saturates, but even then you have excess. Instead of taking 50 molecules, you had 100 molecules, because you are eating likely to take more fat food, and that will push the unabsorbed bile salts onto the colon, and that is a carcinogenic. So unabsorbed bile salt, that is a carcinogen. Absorbed bile acids or bile salts, that is a carcinogen. So that induces the colon cancer path. Okay. Okay. So do you have any question now so far? Here, Sugarland? Or in Cinco Ranch?
I'm sorry, not in Pico Ranch.
Oh, okay. That's fine. So when you do the summary of this class, what we discussed now, we have done the successive breakdown of food. Okay. Successive breakdown of food materials, right? Whatever we eat. Now secretion of enzymes. secretion of enzymes. And we did study hydrochloric acid, we did study HCO3 minus, bicarbonate ion. Also we did study bile salts, okay, and also mucus secretion and water. Mainly the mucus and water that is secreted in the colon as well, okay, also stomach and the small throat, the gastrointestinal tract. And then 3, small intestine, we did study small intestine. What we did study small intestine, and majority of the enzymes. So these enzymes, what we did study on the carbohydrates, digestion enzymes like you have a mono and disaccharides, like a maltose, that's a digested from the starch, right? And lactose. So you have a different transport, monosaccharides and disaccharides. These are the disaccharides. Monosaccharide is glucose, fructose, galactose. So all these monosaccharides, they have their own, transporting mechanism like active transport. And also amino acids, we did study absorption. And then the next part is we did study about the absorption of bile salts, like a BAT, right? Bile acid transported protein. And then finally we did study in the colon part, the water recovery by aquaporin and salt absorption, which is there in the recovery. Okay. So the whole process, we need to have a regulatory process. How this entire process of the digestion is going to be regulated. Do you follow that? So that part is our next lecture of this one, which is part of the digestion 3. Oh, okay. Yeah, I'm just going to the next part of my lecture, on the how this part is going to be regulated. Okay. So this part is just like I mentioned before, it's sensory as well as the hormonal input. So that's what we need to finish this digestion part. Okay. Have you finished?
Yes, sir. Thank you.
Okay. Now we are doing on digestion, lecture 3, and this is for the part of regulation. The whole part what we did study earlier, and the regulation part. We put that in the mouth, okay, and then I just put a drawing like a stomach, okay, and then this diagram you have the pancreas, and then gallbladder, and then liver, which is coming over here, and then you have a small intestine which is going over there, and then ileum, and then you have the colon. So these are some of the part which is just to be mentioned before, but it has been regulated in a very systematic way. Okay, so one is mouth, okay, and then it's going to stomach, and then we have a pancreas, P for pancreas, and G for gallbladder, where the bile acids, and liver, and these are the small intestine. And here this part is duodenum. Okay, and the mouth. Now the regulation of the secretion is going to be sensory. So whenever you get a smell of food, and you pass by, somebody is doing a barbecuing stuff and a burger, and smells of the charcoal and flaming burger, everything, you get your nostrils and smells. Hey, it's good food, right? So that kind of stuff coming on the sensory thing from the mouth, and secretion of your saliva and everything which is coming out. So that is a part 1. And at the same time, that sensory input also increase your stomach. The stomach, in turn, the parietal cell hydrochloric acids. You follow that part? HCl production. Now, let's go on the mouth. Okay, mouth is sensory. Taste of food, that also increase. This is purely of neuronal, okay, neural activity. That will activate salivary gland. plus, I put plus for activation, activates salivary gland. Okay, and it's also taste, also smell. Taste of food and smell of food that activate the sensory, there's a neural part that activate the salivary glands. When it goes onto the duodenum, the food is entering the duodenum, that inhibits the stomach digestion. So that's another regulation. That's a negative one. So food here, smell and everything, taste is secretion of all the salivary gland in the stomach, and then in the esophagus into the stomach. You get hydrochloric acid, sensory output. So you get hydrochloric acid secretion, everything, and digestion, and after that it's pushing onto the duodenum. As soon as the food is entering the duodenum, what will happen? It switch off the stomach digestion. All the hydrochloric acid and all the other secretion of the gastric juice will be put it off as soon as it enters. So that's the neural from the nervous system stops over there. Okay. So how you get the activate of the salivary gland? Even thought of food. Thought of food. Okay. So you get a salivary gland secretion. So that's on the neural aspects or the neuronal input. Now we'll go on to the next part, how this happens. Okay. The stomach, what happened in this stomach? We'll go on to the next part. How the stomach digestion is regulated by the neuronal aspect and the other aspect. Okay. Number one, sensory input that will activate, plus for the activation, plus mouth is an activate, and also it do stomach to activate in the stomach. Okay, the mouth, well, that's again the neuronal aspect, neural input that also activate in the stomach. Stomach means what? It secrete more of hydrochloric acid. You follow that? And gastric juices. And then the stomach, food in the stomach. If it the food in the mouth, that activate into the stomach. If you put the food in the mouth, that activate the secretion of hydrochloric acid. Now, smell of food, that increases both. Okay, it activates. And then you grab a burger or grab your food onto the mouth, and then that will get into the stomach, will secrete the more of acid. And then when the stomach is there, the food in the stomach also activate the stomach secretion. These are all the neural activity. Now, after the stomachs, it enter into the small intestine. The intestine, okay. If the presence in the food, in the positive, I put plus, food, in the intestine, that will lead to block the secretion of stomach. That's a negative. Stomach. You understand that? If a digested food here, it will activate, the stomach. But after this digestion in the stomach, it pushed to the intestine, and the intestinal, if the food receives the intestine, then that signal the stomach here to stop. Hey, there's no more. The food is there in the intestine. I don't want any secretion. Go and stop it. So all this stomach secretion, hydrochloric acid is being stopped, or gastric juice is stopped in the stomach. So it stops there. But at the same time, it will activate the intestinal juices secretion that is in the intestine. Intestinal juice. Intestinal activation, food. Okay. Then once this is being done on the intestine, there is no food in the intestine. I put negative. That meaning no food in the intestine. There's no food. It's been pushed to the colon. What will happen here? Then it gives the positive to the stomach. So that's why you get hungry again after 3-4 hours, right? Stomach, it activates or reactivates stomach. Reactivates. And there's no food that inhibits the intestine. Inhibits, the negative, inhibits small intestine. Intestinal juice. Here what I'm calling all the secretion, okay? Stomach secretion and intestinal secretion juice. So if there is no food in the intestine, then what you feel after 3 hours of your food, where you feel, hey, I'm hungry again. Why do you hungry when you eat? Yeah, I ate breakfast 4 hours before, not now. What happened to you? That food is being digested and pushed to the intestine. You didn't feel hungry at all because that stops the stomach secretion. Now it's pushing to the colon. What will happen to the intestine to colon? The intestine is empty. Now it signals the stomach here, and stomach is reactivated secretion of acid. You follow? So that's why you are getting them. So all this process by neural control, neural mechanism, neuronal or the nervous system under the control of all. Okay. The next part, which is the stomach, how this neural control and sensory and hormone. Here, I'm going to get into the hormone. Suppose in the stomach, how the stomach induces the stomach, okay? In the stomach, when the food which is coming in the stomach, so F for food, okay, food. So it activates the gastrin hormone. Gastrin is a hormone, and then that gastrin in turn again activate. So that is the process which I want to mention. Stomach, food in the stomach that activate the secretion. So if the food in the stomach that activate the gastrin secretion, and the gastrin again will activate the digestion of food or the secretion of the hydrochloric acids and everything here. Okay. So the stimulus for in the stomach is what? Stimulus is neural, N-E-U-R-L, neural control, and also what type of neural here? Vagus nerve. Vagus nerve. And also sensory thought. You know, the people who are so obese and they want to have a surgery. So one of the surgery they want to perform is to cut out the vagus nerve. If they cut out the vagus nerve, what will happen? The stomach activate the gastrin, that process is stopped, and thereby they won't eat much. They always like a grinding mill. They eat, wherever they go, eat, because the secretion of the gastrin, they are so hungry. So if you stop that hunger by cutting out the vagus nerve, like a small surgery on that, then they won't feel that much hungry. They won't eat much, and thereby they can reduce the weight. So that's one of the surgery. They used to do it on sensory. So the sensory thought, you have to avoid sensory thought too, right? And then sensory of food, or the smell of food also secrete. Okay. And the food mechanically in the stomach, just by the weight of food, weight is the mechanical weight of the food in the stomach, that secretes this one. So that's the food, and then the mechanical weight, okay, of food onto the stomach. Something is getting into the stomach, that also induces the secretion of acid. And then what chemical activation here? Peptidases, that's activating the peptides and the enzymes, chymotrypsinogen, trypsin and everything, and they activate via receptors. So that as soon as food comes into that, the receptors will be activated and the peptidase enzyme will be secreted. Okay. Now, where this occur, this gastrin? So the endocrine cells are on stomach wall. I'll draw here. Okay. Endocrine cells, that's the hormone secreting or the gastrin secreting cells on stomach wall. Okay. Suppose this is stomach and you get like this. Okay. So if you have the stomach wall, you get all these cells which is responsible for secreting of the gastrin, and they have gastrin. Okay, cells in gastrin gland, they are present here. And also in the stomach wall, you get the increased amount of mucus secretion, Also, you know about the parietal cell, which will produce H plus and Cl minus, and also it will help in the pepsinogen release. Pepsinogen release. Pepsinogen or pepsin, that is also a fat, a protein digestion enzyme, right? Pepsinogen is a proenzyme, is an inactive form, and the pepsinogen become pepsin, not Pepsi, is a pepsin. So that pepsin is being produced in presence of hydrochloric acid. Okay, so this is regulation which is taking place in the stomach. Okay, do you follow this part? Yes, secretion of this one, and then on the endocrine gland, just we want to see that what happens there. The endocrine and exocrine gland, I want to remember that. Endocrine, what is endocrine? I think you have done before, but I just... Okay, yeah, endocrine gland. Sorry, yes, go ahead. Previous slide, this one. Yes, go ahead. No question?
Question?
Yes.
I have a question. The bleach before that, and you have the secretion of gastrin.
Yeah.
Is that a positive or negative? Is that a positive or negative?
When the food,
regulation?
okay, when the food is there, the gastrin is positive. Gastrin production is more. Okay.
Oh,
Okay.
The food's there.
Yes.
Okay.
And the smell or the weight of the food and everything that activate the gastrin production through vagus nerve. If you cut out the vagus nerve by surgical meaning you are not getting gastrin, you are not getting that much of gastric acid juice. It's only the regular secretion, okay, but not through the thought or the weight or any other thing. That's why they want to reduce, they want to suggest the surgery for the obese people. Okay.
Thank you.
Okay, great. So endocrine gland, that the definition is secretes, these cells secretes the hormone, right? Secretes the secretion of the hormone inside the body. That is the hormone secreting gland. Hormone secreting gland. Hormone secreting gland, otherwise call it endocrine gland. They secrete directly into the body or to the cell. The another one, what we have to remember, okay, is the exocrine gland. Exocrine gland. What is exocrine gland? Secretes outside the body. Outside the body. Meaning it is not inside the bloodstream directly, but it is the lumen of that. That is example, salivary gland. Salivary gland is saliva secretion outside the body. And then gastric gland, which is produce more of gastric juices, and that is gone to the intestinal lumen or the stomach lumen to digest the food. So they are exocrine gland, okay. So they are present in like a liver as well as in pancreas. So liver and pancreas, they have both, both they have a endocrine gland as well as the exocrine gland, okay. And the pancreas endocrine gland, when you know very well insulin, and the insulin is a hormone, and the glucagon is also a hormone, which is present in the pancreas, endocrine glands. Okay, and the exocrine glands, as you know about the liver enzymes, and pancreas also exocrine gland, like a lipases and other enzyme as exocrine glands. So this organ, liver and pancreas, they have serve both purposes, endocrine gland and full of exocrine glands. So the hormonal control that is taking place with the help of liver and pancreas, and thereby it secretes different hormones to help for the digestion. Okay, now we'll go on to the next one, as the duodenum, where you find the liver and pancreas, they place a major role. Let us see what happens in the duodenum. Duodenum or duodenum. The stomach content. Stomach content. What are the digestion food particle in the stomach contents? that activate, okay, whenever the food in the stomach that activate the intestine to say, hey, there's a food in the stomach, are you ready? So it commands the intestine, and the intestine receive the message, and that are the hormonal impact from pancreas and then gallbladder, okay. And when the stomach content enters in the small intestine, it's been activated, the juices and pancreas and gallbladder is activated, and once it's entered there, what will happen? The stomach secretion is shut down. Stomach secretion shut down. Meaning when the duodenum, the food passes into the duodenum, then immediately this one will shut down. Stomach secretion, hydrochloric acid, gastrin, everything is going to be shut down. So it decrease output, and thereby it's only in the small intestine is going to be there. Okay. The hormones which is playing a major role here in the small intestine or duodenum, hormones and the secretion of secretin. This is the one hormone called secretin. Okay. Another hormone called vasoactive intestinal peptide. Vasoactive intestinal peptide. This is another hormone. We call it VIP. Very important peptide. No, very important person. No. VIP is vasoactive intestinal peptide. VIP. Another one is the cholecystokinin. Cholecystokinin, otherwise called CCK, another hormone. Then another hormone is gastric intestinal peptide. Gastric intestinal peptide. Otherwise GIP. Okay. So they secrete. Once they secrete, and they can help onto the dual function hormone, like dual function hormones. I'm coming there. Yeah, I'm coming up now. Dual function hormones, and this will it will activate the small intestine. Okay, it activate. Okay, and the secretin which will activate here onto the specific thing. It will help the activate the vasoactive and the cholecystine and everything. So this hormone, list of hormones, secretin, vasointestinal peptide, and cholecystokinin, gastric intestinal peptide, all these, they form a dual function, meaning one hormone, it activates when the food is there. When the food is not there, it is inactivated by itself. Okay. So the secretion of the small intestine, it's been controlled by hormone. So intestinal secretion is controlled by secreting VIP, GIP, CCK, cholecystokinin. So in our part earlier, which we studied with the absorption and digestion, and now we are talking about the regulation of the secretion of the juices and hormone. The regulation by mainly the hormones. Do you follow that part? How this hormone is being regulated, what hormones, and where it regulates. Okay. The duodenum. Okay. Now, again, the following the duodenum again. Duodenum, stomach input, when it drops, stomach input drops, right? I put a double arrow because it food enters into the duodenum. As soon as it enters into the duodenum, or the stomach input is dropped, there's a hormone called enkephalin, 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 okay, that is released. Okay. This after this release, that lead to increased in pH. That's why you have a neutralization of acid. Okay, increased in pH, and also it increase. Okay, it block the increased secretion of gastric juice production. Okay, gastric, it blocks here gastric juice production, because in the stomach full is dropped, so there's no, so the enkephalin will stop the stomach gastric juice production. Okay, and also it increases the enteroglucagon that is coming from the carbohydrates. As soon as the carbohydrates enter, because the food is entering into the duodenum, carbohydrates, okay, that activate enteroglucagon, another hormone. Okay, so the food, a type of food, and there's a mechanism which a different type of hormone which will do both. One is activation of the intestinal juices, and also it inactivate into the stomach. Okay, so that's the part which is happening into the duodenum. That's, you should follow that. And then we will go on to the hormone release process, which we discussed earlier, like a hormone. These hormones release, how the hormone is released earlier, which we studied by exocytosis. Okay, suppose if this is a cell and you have got some signaling molecule, the signal which is coming from the hormone, like signal, either from neuronal output. So you have a different process. They activate different molecules in a biochemical way, like a signal transduction, and finally these vesicles where you have the hormones which is there, and this is being released by exocytosis. And this release is being regulated by receptors. Specific receptors are there. Here, these are the receptors. We call it the signal coming and play into the cellular receptors, and then receptors activate step 1, 2, 3, 4, activates and molecules, and then the signal transduction molecule release this by exocytosis, and then the hormone is released. The hormone release is a very complex biochemical pathway, but it involves the receptors. That's what I want to remember. If you block the signal, or some sort of a duplicate signal, or the receptor antagonized, that's some other drug, and this formulation will block these receptors, and thereby you can block the hormone release, and thereby you can regulate the secretion of the intestine. You follow? We studied digestion, absorption, and digestion helps, you need secretion of enzymes and hormones, and then how the hormones are released, that depends upon the receptors, and that receptors, in other words, is being activated by signals. So if you want to stop the hormone secretion or the enzyme secretion in a high level, so you have to block the receptors. So that's what the pharmaceutical industries, they are looking forward. Okay. So the hormone action of the peptides. So where this hormone will, what the hormone will do. Now the next step. Call it as a hormone action. Okay. The hormone, suppose it is a peptide hormone. There are different type of hormones, but the peptide hormone, peptides, what you have to receptors, the protein, as I mentioned before, receptors. The hormone receptors are there, and they are called receptors protein. Specific receptors for specific hormones, like insulin receptor, glucagon receptor, thyroxine receptors. So individual hormones, you have individual receptors, and these receptors nothing but the protein are the target cells. Receptors are the target cells. target cells. Receptors are present on the membrane target cell. Suppose this is a cell, and this is you have a hormone receptors. Hormone receptors. So the earlier one which I mentioned before, here in this one, how the hormone is released. Now the hormone is in the blood pool or in the blood. Now this hormone secreted by exocytosis mechanism. Once the hormone is swimming around or floating around in the blood, then where it will act, the target for the hormone is the cell, and the cell is the nucleus. The cell will have a hormone receptor. It welcomes the hormone, respective hormones, and thereby it induces signals. The hormone binding with the receptor induces signal, and it includes, activates, the hormone receptor activates intracellular transduction, signal transduction process, that especially G protein, and this G protein which is coupled with the membrane level, and they are act as a second messenger. The first messenger is the hormone, and then it binds with the receptor onto the targeting cells, and thereby it activates the G protein coupled receptors. We call it as a G protein as a second messenger. And then this cell, what it will do after the signal, it activates the gene transcription process, and thereby it secretes or it synthesize proteins, and that protein from a gene, and that synthesized the protein is either an enzyme or a hormone again, or another hormone. So this enzyme has been used for the digestion process. Do you follow now? How a hormone, which is a secretor, signaled by the taste or food or anything, mechanical receptors, so it will secrete. The hormone secretes. The hormone is going on the targeting cell. For example, in this case, parietal cell, the hydrochloric acid output, or the transport activator, or the enzyme level, or the salivary, what is called the saliva secretion, or the intestinal enzyme, or if you go on to the gallbladder, and then pancreas, and to lipases, all those enzymes need some signal. Where this signal coming from? That's coming from an activating hormone. So there the hormone is coming from another cell. So that's what I'm giving a overall picture how one is related to the another, and thereby it regulates.
Dr. Sullivan,
Yes.
what's below second messenger?
The second messenger is again the synthesis of proteins. Second messenger that will help to synthesize, activate the gene which is in the nucleus, and that is specific for a particular enzyme, or specific for another protein, or specific for another hormone, that will help. So I told you, whenever you think of a food, you secrete saliva. Think of food, you have hydrochloric acid and gastric juice production and the enzymes and everything, correct? So if you think, you get it. How? It is neuronal control. The neural control getting into the hormone, and the hormone activate those enzyme and secretion of those juices, everything. So that's what I want to make sure, the pathway of secretion. Either it is saliva, or in the mouth, or in the stomach, or in the small intestine, or in the colon, or anything. So how it is being totally regulated. Okay, the overall picture here, what we have studied so far, as a work, right? Work to be done, right? And that's process on the heat. And then you got the feces which is coming out after the digestion, and the energy which we did study about the food. So food enter, and then work done, and then you got the heat is being produced. What we have done so far, it is the intestinal regulatory process. Intestinal regulatory process. Regulation, intestinal regulation. And you have to go on to the environment, and then the behavior and all other aspect we will do on the next part of our lecture, how we will study the process of circulation, muscle contractions, and then hormone release and everything in a specific way. So that's we will go on to the next part. Okay. probably the next week of this lecture. All right? Do you have any questions so far? So just I want to conclude this digestion part. Will be over with the digestion part 1, 2, and 3 of the lectures. Sorry? This is chapter 15, and also it's my lecture on class notes. Okay.